Okay, welcome everyone to the seminar. Welcome, Diego. Thank you very much for accepting our invitation. Yes, close the windows. Thank you. Diego Fontaneto is a researcher at the Water Research Institute in Verbania in the north of Italy, which is part of the National Research Council of Italy. He started off as a PhD student and later as a postdoc at the University of Milano, after which he went on to the Imperial College in London. And then he did 2 years as a research fellow at the Natural History Museum in Stockholm, after which he went again to the Imperial College in London. And in 2012, he started at his current research position in Italy. Throughout your research history, I guess the most consistent topic reoccurring all the time are rotifers, whether it is taxonomy or their ecology or their distribution. But he also did a lot of research on more broader topics like biodiversity patterns of rotifers, but also other organism groups, and more biogeographical patterns, which we will hear about today. And if I scroll through your Google Scholar records, I would describe you as a very creative researcher. I think whenever he gets an idea of some topic, whether he has done research on that topic before or not, he takes up the topic and does maybe 1 or 2 studies on it. And one of those studies is, my favorite. My favorite one of them is a study where you looked at rotifer species growing on lichens that grow on different trees, and you looked at whether the species occurring on different species of lichens were kind of related to that substrate species. So just to give one example. Thanks again for being here. I'm curious what you will tell us today.
Yeah, thanks for the invitation.
You're welcome.
And if I have to add something that is common in my scientific career is rotifers and traveling. So I like traveling, and rotifers help me to do that. And by traveling, I wanted to work on biogeography, of course, because you go in different places, and then it's possible to understand distributional patterns of animals. And I started as an entomologist, and biogeography in insects is totally cool. So I wanted to translate biogeography of insects into rotifers, looking for endemic species and strange distributional patterns that could have an explanation through the history of the geology of the place. But working with microscopic animals, I soon discovered that it was not really possible. And people already knew that. So that if you look at textbooks in biogeography, there's always sentences like, microscopic animals, don't look at them. Already Darwin and other travelers, when they were traveling around the world, they were sending back home samples of mosses and lichens and soil to look for microscopic organisms. And there is a letter by Ehrenberg, who was one of the protists, microbes, meiofauna people at the time, who wrote Darwin saying, stop sending me mosses from wherever you are in the world, because I find only the same species that I have in my back garden. So I stop wasting my time for that. And this developed into a more scientific activity like scientific field. And the name of what people thought about biogeography and microscopic animals and microscopic organisms is the sentence, everything is everywhere. And I didn't put it in the title because let's say that everything is everywhere is a hypothesis that people said you cannot even test it, so you cannot really prove it if it's correct or not. And what I will try to discuss is more why animals can be different from larger animals, and I will forget about bacteria and tiny organisms, and more focusing on like which are the biological traits that may favor broader distributional ranges, and not only size, of course. And to do this, I start by introducing nothing because it doesn't work anymore.
It was working so nicely.
Yes.
Maybe plug it on then.
Yes. problem is that it doesn't even go out of this. Okay, good, found the solution. So we start with these definitions of what is biogeography, and this combination of ecological and historical factors that drive speciation, extinction, colonization drift with the historical factors. Together with ecology, they drive where we find organisms at the moment nowadays. And that's true for everything we see in the surroundings, including people. And for some groups of animals, we know a lot. So we know where they live. We know the phylogeny of these species living there. We know fossils. We know ecological models to drive distributional patterns. So we can make inference on why do they live where they live now, and they don't live in the places where we don't find them now. We can understand whether they were extinct there or whether they never colonized that area. We have a lot of information. The same for obscure animals like velvet worms, but for which we still know a lot. We know where they live. The different colors in the world represent two different groups of these animals. So phylogeography has also a phylogenetic structure in this case, with the two main groups having two different distributions. And people worked on this for ages and ages and ages. And you had a talk by David Nogues recently, and he works on several of these topics, putting strong mathematical background and putting empirical data, mathematical analysis, treatments of the data, and some hypotheses. People develop lots of new theories on distribution of organisms. So we have island biogeography, that is the god of what we know of biogeography, with an engineer talking with a biologist long time ago and discovering that there are patterns that we can analyze in nature. And then from island biogeography, we have any possible pattern in the world has been studied. There are entire journals that are devoted to this distribution. So we know that biogeography exists. But then when we go to tiny microscopic organisms, they become not interesting in principle, because people assume that they are small, and being that they are small, they survive even if you take them out of their habitat, so they can be moved all over the world. They have also higher productive rates, and they colonize new areas continuously. And I focus my talk now on a group of rotifers. They are tiny animals, meaning that they are 100 micron, and the giant ones are 1 millimeter. But the ones that are 1 millimeter, they are thin and long. So they are still very, very small. Even if on one direction, when they are fully extended, they can be 1 millimeter, but they are much less than that. And on this group of rotifers, biogeography seemed not to exist, but also ecology seemed not to exist much. So like there was this study in Belgium with some letters that don't come out, but doesn't matter, in which the idea was to study a gradient of urbanization, to see whether a city is different from a rural area or a natural area. And we looked at different groups of organisms, and we can see that some of them responded by diminishing body size at the community level. Some of them, the top two, responded by increasing body size at the community level. So butterflies, moths, and grasshoppers. And Belloid rotifers, the third one there, they didn't care. As long as there was a lichen on which they could survive, the species that live in lichens, they live exactly well without changing anything of their community composition, at least regarding body size. And they are the only group, together with orb-web spiders, that they didn't seem to respond to urbanization. And this is a pattern that will recur over and over again in the talk, that rotifers seem not to care much about geography, but also about ecology. So of course, if I look at mosses, I find the species living in mosses. If I work on lichens, I find the species living in lichens. The ones living in freshwater are freshwater. But as long as freshwater is available, they are there. It can be a spring higher up in the mountains or a pond in the middle of a desert. If water is there, most of the species live there. So they are really broadly ecologically, not only geographically. But now let's go back to this slide that we have already seen to see why we think that microscopic organisms don't have biogeography. And the reasons are mostly these three. The main one is tautological. Small organisms don't have biogeography because they are small. And that's true, that they are small, I mean. And given that they are small, they are below a threshold for which the surface to volume ratio allows water to flow continuously without any problem. So if they are in water, they don't have any circulatory system because water and everything passes through them. Because surface is so much more than volume. For larger organisms, you cannot do that. And that's also why larger organisms can sustain life outside water. If you are super small, only water is allowed to you. Because as soon as you are out of water, you dry out. This has a consequence that if they live in water and when water is not available, they dry, they invented any possible way to survive without water. Because if they live in a puddle in the field, but the puddle gets dry, either they die all and gone forever, or they need to find a way to survive desiccation. And they do it all the time. There are different ways to do it. Some organisms produce eggs that are specifically devoted to do that. Others build a case around them to protect them against water. The group of rotifers I'm working with, and I will tell you more later, I told you already that they seem not to have biogeography, they seem not to have ecology, and they don't care at all about lack of water. So if you have them in water and you dry them, they dry. They wait until water comes back again. They don't do anything apparently. They simply shrink, collapse all the body parts, and when water is back again, they are there again. This adaptation is so strong that you can freeze them. If you want to lab culture and you want to go on holiday, you simply put them in at -80 directly, and after 2 months of holiday, you come back, 5 minutes in water, they are happy as they were before. And not one dies at -80. So apparently, the colder it is, the better it is for them to survive something that is impossible. And even on the other side of the temperature range, one way to fix them is to boil them and then put some chemicals to fix them. If you are not quick after boiling them in putting chemicals to fix them, they start moving again. And everything is thought to be related to this ability to survive desiccation quickly and immediately. And as a byproduct, they survive total frost, boiling. They even survive ionizing radiation. They acquire science fiction features simply to allow them to survive lack of water. But given that these resting stages are really super resistant, and they are small and super light because they are dry, they can be dispersed very easily by wind, for example, or by birds. And there are histories of people who take ducks, put ducks in water, shake them to see which propagules of tiny animals come out of them. And there was also a biogeographical meeting some years ago when people, as soon as all the attendees went back home, they received an email by the organizer saying, now, we didn't tell you anything before, but take your boots, take a toothbrush and brush them. Send us the dirt that comes out. We will tell you that you dispersed to your country the tiny organisms from our country. So you were making invasive alien species all over the world, which is true, but then doesn't happen so much because most of the resting stages survive a bit, but not too much, except for the ones of rotifers that we will see how long they can survive. And then, of course, all these organisms, animals, but also protists, they have a third feature. That is that they may not need sex to increase population size because they simply reproduce asexually. So if one single propagule arrives in a suitable habitat, they start producing offspring, and then they become millions very quickly. So they are biologically pre-adapted to be without biogeography. And these are the organisms I was talking until now, on which I will focus a bit. Rotifers, my focus, but also some tiny nematodes and water bears, tardigrades. And some groups of protists seem to do the same. And they are, what they have in common is they are small, they survive desiccation, and they have these strange reproductive abilities to separate, increase on population size, and reproduction with sex and males. What are the problems we have? Is that most of the literature on survival resistance is based on anecdotal evidence. Like, as I did before, I take my samples, I put them at -80, I take them back and I see, well, they survive. But now we know a bit more. So we know that, for example, looking at rotifers, nematodes, and tardigrades kept in different species of lichens in the museum in Stockholm, I was very happy to be able to look at the samples that were collected around Linnaeus time. And over there in Sweden, they have this monitoring program for which some of the common species, they are sampled all over Sweden every year, and they are kept in the museum. And they took small bits of these lichens, hydrated them to see how many animals could recover and how many could not recover of these three main groups. And we found that lichens kept 1 year in the museum, most of the animals survived well. But then as soon as lichens were kept more than 5 years, most of the animals were dead. So it's true that they survive up to 5 years, dry in a museum, but they don't really survive so long as one would expect. So my idea was to have a fantastic paper saying, these are the rotifers that Linnaeus collected, and they are still alive, but we didn't manage to do it. And we discovered that it could still be potentially true, but around this time was the last time that they fumigated the museum with chemicals to prevent bad things to happen to the lichens. And it's true that the rotifers can survive desiccation, but if you put bad chemicals on them, they die. So maybe our result was not due to the actual survivorship of the animals, but was due to chemistry. Anyways, surviving years for an animal that have a life stage of 20 days is still long enough to be dispersed. And for biogeographical consequences, surviving a few years is still a lot. But then this paper came out this year in which they took permafrost from Siberia and they found living animals in the permafrost. Well, not in the permafrost, when they melted the ice, these animals came out. And it was very controversial. They need to do lots of tests to convince that they were really from the ice. And the most convincing one was that none of these species was found in the surrounding. So it's something that doesn't live there anymore. It lives, actually, it's a new species that nobody has ever seen before. And it's found only in that layer of permafrost, and they found more than one resurrecting animal. So if this is true, 24,000 years survivorship dry or frozen is long enough to be moved wherever the wind may take you. So they survive resistance. The other problem we have is if I find the same species in Italy and Switzerland, it's okay, can be the same species. If I find the same morphology in Australia, is it the same species? Or is simply that taxonomy in organisms that are so small, if I find a round bubble with two spines, I give it the same name because there's nothing different. And actually that's what happens. So do we identify reliable units of the diversity? Or it seems that we are unable to do it? With morphology, even with advanced microscopical techniques, we still don't see the true differences between species. Well, we have DNA to help us, and until now, all the studies using DNA to identify species or to support species identity work well. So the same species from morphology in Italy and Australia invariably comes out as really the same species. So the ones that are globally distributed and are found all over the world, DNA suggests it's the same species. There are problems, like the genus Rotaria is a genus with tens of very easily identifiable species, and they are all supported by DNA. And then when you have a species that belongs to the genus Rotaria, for sure, but you cannot identify it as one of the known species, you give it this name. So it's the garbage bin. When you use these dichotomous keys and you arrive at the end, no feature to identify it, you give it this name. So taxonomists already knew that this one was problematic. It's not one species for sure. And only in the UK, DNA tells that there are maybe 30 species. And it makes sense because this is really the queen of the ecological variability. It lives in salt water, open ocean, brackish, sewage treatment plants, oligotrophic ponds, mountain spring, glaciers, Antarctica, wherever. Wherever you go and there is water, it's there. And of course, it's not one species in this case. But we already knew that as taxonomy. So I will not base much of my biogeographical inference on the morphology of this, because of course it's everywhere, but it's hiding things. Another species that was everywhere is this one. As long as there is salt water, it's there. And it's heavily used by people for culturing fish. So it's used in aquaculture. And 15 years ago, people started realizing that if you sample that species in Japan and you grow it in the lab, it grows very well at 13 per thousand salinity. If you take it in the Mediterranean, it grows at 30 per thousand salinity. Different laboratories were using totally different protocols to keep it alive. And then body measurements were like, this is one species, one of the populations from dorsal, lateral, and ventral. This is another population, and this is another population. Body size is quite different, but often in the same pond, you find a lot of morphological variability. So people didn't really know what was going on until here it was written that we sampled almost 2,000 animals for more than 500 populations. And we discovered that there are 15 species. Nuclear markers, mitochondrial markers totally concur in saying they are 15. And the trouble that people didn't realize it is that in some ponds, you have up to 5 of them living together. So people realized that there was morphological variability, but they were not able to find what was going on with this in the same place, small, medium, and big, living together. Now we know this because they are different species in the same place. So again, here, the result is that we know where they live, we know what they are, we can identify the different species. And it's a common feature in all the tiny animals. Whatever you do with tiny animals, there are cryptic taxa. But when you work with DNA, then you can do integrative taxonomy and identify species. So we know what we are talking about when we put names in species. So we have reliable units of diversity. And now the last one. Do we have data to do biogeography on these animals? So we have data on distribution. And we started addressing this question by gathering what was published for 30 years in 2,000 papers. We didn't go before 1960 because taxonomy for 1960 for rotifers is a bit shaky. So we started when we can trust the names. And that's what we came out in the world. Like darker areas are richer in species and white, no records. And we started building models to understand what was driving richness, well, diversity. And the main driver is how many papers were published in each place, which means that we have no idea about biogeography, but we know very well where rotifer people work or go on holiday. That's the only thing we know. And if you do this with the other tiny animals that live together with rotifers, it's always the same. We don't have data to do biogeography. So that could be a problem why we don't find biogeography, simply because we don't have the data to do it. And now I introduce, the sentence. Everything small is everywhere. If we don't have data to understand and analyze biogeography, can we deal with this? My answer is yes, you can find some tricks. One of the expectations is that if everything is everywhere, then in a small valley, I should find all the diversity of the world, if everything is really everywhere. And looking only at 35 mosses in one afternoon in one valley in the Alps, we found 20% of all the worldwide diversity of this group of rotifers, knowing that half of them live only in lichens, a big part live only in soil, and the ones that are known for mosses, we found almost all of them. Is it true? Well, we publish it as a way to say it's true that everything is everywhere because we found a lot in a small place, in a very, very small sample. But it's also true that most of the known diversity of diploid rotifers comes from the Alps, because the only people who ever worked on rotifers on this group, they worked in Austria, Germany, and they like the Alps. So it's true that I find everything that lives in the Alps in the Alps. It's not so really convincing. But still focusing on the Alps, we looked at different samples from west to east on different kind of substrate, focusing on a very peculiar habitat that is the top of mountains above 3,000 meters. So I had money to do what I like, hiking and collecting samples. And the result was, wherever you go, community composition of rotifers living in mosses and lichens across all the Alps, geography has no effect. So the probability of finding the same similar community half meter far away or 300 kilometers far away is the same. So at least at this scale, for diploid rotifers, there's no geographical effect. And there's also no ecological effect because dolomites are totally different from Western Alps as a substrate. But the rotifers that were living in different species of lichens and mosses, they didn't care. And I also fell in the trap of traveling all over the world to see which species I could find, and actually I found always the same species. What does DNA tell us? The same, that the species are cosmopolitan. And there is this species for which I found exactly the same mitochondrial genotype in UK, Tanzania, Thailand, and Taiwan. So the three countries that start with T plus UK, they have exactly the same haplotype. In the other species, we found a gradient, like genetic diversity within species had a correlation with geographical distance. So there was a pattern going on there, mostly between continents. So when the distances were within continents, geography seemed not to have much effect. Between continents, a bit. So the consequence for us was that, yeah, there are phylogeographical patterns, there is a signature of colonization, larger scales than with mammals or birds or insects, but it is there. And my explanation for this species that was found in exactly the same one in UK, Tanzania, Taiwan, and Thailand. We found it in a very strange habitat, mosses in golf courses. And they are all places where the British went to the golf courses and most likely bringing soil from the UK to do the golf course exactly as they wanted. So maybe we found the same species because it's the British species that was scattered all over the world. But I have no idea, no way to prove it. For all the others, DNA gave evidence of biogeography existing. And the consensus among people working on microscopic animals is that at the individual level, small and big, you have the same side. But then species for larger organisms have a narrow distribution, narrow at least within a continent. For microscopic animals like rotifers, species can be moved a bit higher geographically. So you may have 2, 3, 4 more continents where they live. And then as soon as you go to genus level, most genera are always cosmopolitan. While for larger organisms, you get that with family or class. So it's a shift in taxonomic hierarchies in biogeography. Again, is it true or is it only our systematic way of making categories that is mimicking something that is not different? I don't have the answer. But I finish with saying something else on the other part of the sentence, because it's not that only everything is everywhere biogeographically, but you find whatever if the habitat is the right one. So the environment selects. Is this true for rotifers? Because I told you already that ecology is not really so important. And we start with the flagship species. Flagship because it's so easy to identify. It has spines there that are so unique that even scanning at the microscope quickly, you identify the species. This one lives only in the northern hemisphere. It has never been found in the southern hemisphere. So there may be some biogeography. Another one called Macrotrachela cancrophila. Cancrophila means crab lover. It lives only in the freshwater crab, in the gill chambers, and only in Italy. It has been found twice in freshwater crabs in Italy. It was an endemic species, so I was very happy that Italy had one of the very, very few endemic species of rotifers. But guess how many people look at rotifers in crabs? Yeah, twice. And twice it happened, once in Italy and once in Bulgaria from freshwater crabs from Italy that were taken there and people looked at them and they found rotifers. Then I had the opportunity to go to New Caledonia. The first evening we arrived, a mangrove crab walked to us, and we were there to look for microscopic animals in epibionts on other animals. We looked at that crab, and we found exactly the same species. So the habitat selects. It likes crabs. But in Italy, it's freshwater. And in New Caledonia, it's marine. And the one in freshwater rarely comes out of water. The mangrove crab rarely goes into the water, but it has water into the branchial chamber. So for these animals, it's a super protected environment. If you take them out, they die almost immediately. They are unable, these rotifers are totally unable to survive desiccation, totally unable to survive out of the crab. How they can move from Italy to New Caledonia? Most likely because they really live in every crab all over the world. Simply, we never looked at crabs all over the world. So my project for the future is big, tasty crabs all over the world, checking for rotifers living inside. But going back to lichens, something that Geza already said, if ecology exists, I was expecting that 4 different species of lichens will have different bacterial composition, different chemicals inside them. And then those lichens could be grown on rocks or on trees. And then the substrate can give different things to the rotifers living on them. And then we looked at an extensive sample in Sweden. And what we found is that 12% of the variability in community composition is explained by lichen species. And then additional 4%, well, 6% if you put together also the substrate with the lichen. So overall, 12 plus 6, 18% of the variability in community composition is explained by habitat, which is something, but it's very, very little. It means that 80% is totally, I could not find any explanation for it. And the same if we move to, sorry for the slide, different species living in lakes. Most of the time, what is not explained is well above 50%. Even if you put environmental features or geographical features in different transsurface analysis or moral segment vector mapping, whatever you do, geography doesn't seem to work much. In this case was genetic differences within one species, totally unexplained. And the only thing that was explained by environment in this case was because one group of haplotypes of this species was really, really loving a lot high sulfates. So that was the only ecological pattern for that group of animals. Otherwise, no ecology. The other ecological pattern we analyzed was not environmental, but competition with different rotifers. So that biotic exchanges and not only abiotic environmental features. And in this, we selected a small freshwater crustacean that is common everywhere. And it's a proper hostel for rotifers. If you look on the ventral side, there are rotifers everywhere. But what we discovered is that one species of rotifers really loves to stay here in the middle. Another one stays at the base legs. Another one loves to stay close to the cloaca, one close to the mouth. But the propensity of going where they would love to go changes according to which other species are present on the rotifer. And there is a winning species, the one that stays here in the middle with this density function overlaying on the crustacean. When they are alone or when they are with the others, they stay there and they are the winners. All the other species, if they are alone, they would love all the base legs, all the legs across all the body. But if there are the ones that live close to the cloaca, they cannot colonize the legs towards the back parts. So they really interact among each other, and community composition is a stronger driver of occurrence and distribution than abiotic factors. So apparently for them, ecology exists, but with competition, or at least interaction with other species being much, much, much stronger with clear patterns more than with abiotic features. So I can conclude that rotifers have wider distribution. Yes. They have higher dispersal rates. Yes, they really move a lot. And if you take a net in the air, you find propagules of rotifers. If you put a petri dish out on the balcony with some water, the following day you find rotifers inside that fly in. But still, the differences with larger organisms exist, but they are not so strong as we assume, especially because if you look at the other animals, tardigrades, the story is totally different. Tardigrades, same size as rotifers, same survival capability, maybe even more, same dispersal abilities, same reproductive strategies, same habitats, you always find them together. For them, phylogeny is fantastic. Even within a species are usually restricted to a small area. If you do phylogeny at the genus level or species complex or whatever level you would do, clades are totally geographically separated. And the age of separation of the clades matches geological histories. So these animals can disperse like rotifers, but they don't. Rotifers, they can and they do. I have no explanation why, because they do exactly the same things, exactly in the same way. But they may have some feature that we have no idea what it is that make rotifers be everywhere, tardigrades not at all. Other animals that live together with rotifers and tardigrades, and they are not animals, but they are small, and they are same size. They are these state amoebas. It's a group of people working in Neuchâtel, working a lot on them. And they did this great job on one species that lives only in the northern hemisphere, sequencing all the known populations from Europe and all the Holarctic. And they discovered that DNA has a strong geographical signal. What they said is that Europe has been colonized by very, very few of the very, very little of the genetic diversity that exists in North America. And it's a subset of what exists there. And only few of them managed to come. And there's really a decrease in diversity going from America to Asia and then Europe. So for these organisms, same size as rotifers, same survival capability, same habitat, they do like tardigrades. Geography has a strong effect in driving distribution. I go to the end, and this I skipped because it's just making things crazy. Simply saying that we need to do better taxonomy to know our species, that's totally fine. We need to have better data to be able to identify biogeography. But the question is not whether tiny animals have biogeography or not. The question is what allows some of them to have widespread distribution and doesn't allow others. But most importantly is that with tiny organisms, we can do experimental biogeography, something that cannot be done, well, could be done if you completely wipe out the fauna and flora of an island and wait for things to arrive. But you can make artificial islands and check biogeographical theories using dispersing animals and tiny animals and put in them. And we know that this can be done because for some organisms, biogeography has a meaning, and then it makes sense to compare what happens with the different species with and without biogeography to check expectations on experimental biogeography. Okay, with this, I conclude showing how beautiful they are, the genus Rotaria. Thank you.